The overall goal of this experimental procedure is to develop a preclinical mouse model to study the role of extracellular vesicle-mediated communication between tumor and mesenchymal stem cells.
This method can help answer several key questions in the cancer biology field as to how tumor and mesenchymal stem cells communicate by means of extracellular vesicles and whether these support cancer progression.
The main advantage of this technique is that it allows to study the role of mesenchymal stem cell education by cancer EVs on the progression and metastasis formation in vivo.
The implication of this procedure extends toward therapy of cancer because we can use the assay to interfere with EV communication and downstream signaling to inhibit cancer progression.
Besides providing insights into the EV-mediated communication in osteosarcoma, this method can be also applied to many other cancer types. Indeed, bone marrow-derived mesenchymal stem cells are readily recruited to tumor sites where they can differentiate into cancer-supporting cells.
Begin by seeding 8 175 square centimeter flasks with 3,143,B cells each. Culture the cells using EV-depleted medium for 36 to 48 hours. When the cells are 80 to 90% confluent, collect the supernatants from the 8 flasks for EV isolation. Next, use differential centrifugation to remove all of the cells and cell debris from the supernatant.